Today we are going to discuss about what is the t-arm PCR. It is a type of polymerase chain reaction which amplifies a specific DNA sequence. T or MS means Tetra Primer Amplification Refractory Mutation System PCR. It is a type of PCR which amplifies a specific DNA sequence only. Like we can see in this figure that there is present a T nucleotide at a certain position. A T-arm PCR designed for this DNA sequence will amplify those DNA sequences when they have T nucleotide at this position. And when T nucleotide is not present at this specific point, then those DNA sequences will not be amplified. So T-arm PCR is a nucleotide specific PCR. And the basis of T-arm PCR, we can differentiate between different DNA sequence at nucleotide level at a specific point only. Suppose we have three individuals. They have three different genomes. For a certain gene, we can see that individual 1 has allele A at a specific point, the other individual has allele G, and the third individual has nucleotide T at this position. So those mutations are called SNPs. So SNP means presence of different nucleotides in different individuals at a certain locus. We can see this DNA sequence from two individuals. One individual has a C nucleotide, the arrow, the pointed Y highlighted by the arrow, and the other individual 2 has a T nucleotide at that point. So these two individuals have difference in their genome of single nucleotides, and they are called to be SNPs, single nucleotide polymorphisms. If we look at this figure, we see that the individual 1 has a C nucleotide at position number 9. And the forward strand. This individual has C nucleotide at this point. Now let us see another individual. This individual possesses nucleotide T at that position number 9. So they have different nucleotides at position number 9. Comparing these two individual DNA, we can highlight the difference at nucleotide level between these two individuals. Why we want to carry out a t-arm PCR? We want to carry out a t-arm PCR because we want to know that how many individuals have a certain nucleotide in a population. We want to explore the presence of nucleotide, the frequency, the percentage of a certain nucleotide like A, T, C, and G in a population. And then we later want to know whether this nucleotide presence is associated with a disease or not. Or this nucleotide has an effect on a certain disease. That effect may be enhancing the disease or preventing this disease from occurring. So for that reason we carry out t-arm PCR. We carry out it because it is simple, repeatable, and economical. It is used to find out the frequency of an allele in a given population. Frequencies mean the percentage of an allele, the ratio of an allele in a population, and that frequency is later associated with in certain diseases like coronary artery disease, cancer, diabetes, etc. Or it can also be a protective allele against a certain condition or disease like lung cancer, breast cancer, etc. T-ARMS PCR provides us an easy, reliable, and it is most important, it is cost effective and it is easy to carry out. For find a frequency of single nucleotide variants in a given sample size. Suppose we have 100 individuals, we have 1000 individuals, and we want to know the presence of T allele in this population. Then we will design primer specific to T allele and will screen whole of the population for the presence of them, and then we will know that how many individuals have T allele, how much percent of the people have T allele at their specific point. Actually, T-ARMS PCR implies 4 primers to amplify DNA sequence. 2 of these primers are unique and are called inner primers. The other 2 primers are just conventional primers used in other conventional PCR. The 2 unique primers are inner forward and inner reverse primers. They are named as inner forward and inner reverse, while the 2 non-specific primers are known as outer primers. There are also 2 outer forward and outer reverse. Let us consider the human microRNA 585 gene. This is the human microRNA 585 gene. We can see, pointed by the arrow, that this gene has a C nucleotide present at a specific location in this gene. The yellow region is composed of the gene. Now we can see that another individual has a T nucleotide at that specific point. So these 2 individuals have different nucleotides in the microRNA 585 gene. Now we have designed 4 primers, 2 inner primer or 2 outer primers for this microRNA 585 gene to find out the variants present in this gene. We have designed a forward primer. This is not unique primer and it is attached to the DNA molecule and will give us a product of 202 base pair. If you want to design primer, then there is another process for it. We will design primer on primer one software which is available online, and that it is very easy to design primers. When you design primer there, then we can apply this primers for carrying out the PCR. The reverse primer is C is specific for the C allele and it has a length of 297 base pair. The primer has not a length of 297 base pair, but the product produced by this primer will give us a 295 base pair product, and this length will be identified after it is run on agar-agar gel in the presence of a DNA ladder or a gene marker. We have 2 other primers for this gene, the forward outer primer and the reverse outer primers. When these primers amplify the DNA sequences to which it gets attached, it will give a size of 446 base pairs. On the basis of these length of the product size, we will identify which primer has worked. When we see the 446 base pair DNA, we will say we can conclude that outer primer's product is formed. Let us see how this primer works. First, the DNA will be denatured in the PCR. When the DNA is denatured, it will be separated into 2 separate strands. Now the DNA, the primer we have designed will try to attach to these separated DNA molecules when the temperature for annealing reach to annealing temperature. So this primer comes and tries to get attached to the DNA separated molecules. The reverse primers get attached here. Because these sequences are complementary, so the attachment is successful and polymerization will be carried out, and this primer will give us a product, and we will be able to recognize the product on the basis of its length, because we know the length of the product of this primer. So when that length is obtained, we will see that this primer has worked. The reverse primer has worked, and the C allele is present in the genome. Similarly, we have another DNA molecule. This will be denatured. When it is denatured, it will be separated into two DNA strands. When the strands are separated, the primer will come and try to attach to this DNA. So the forward inner primers comes and attaches to this DNA molecule. This is DNA molecule of a second individual. It means that there is presence of T nucleotide at that point, and the primer will get attached and the product will be formed, and we will be able to recognize the product on the basis of the length of the product. So this is how the inner primers of the ARMS PCR works. Now let us see further in detail. This is a DNA sequence having a T nucleotide at a certain point. This is the forward strand of the gene of an individual having the T allele highlighted in red. This is the other strand of that individual. It must contain a opposite to the T, complementary to the T. The primer comes and gets attached to the reverse strand. So the forward inner primer will get attached to the reverse strand, and it will be amplified. One thing we should remember about the PCR is that the strand to which the primer is attached is not amplified, but the other strand opposite to their strand will be amplified, because the primer attaches complementary nucleotides to the growing strand using the template. The reverse strand as a template, or the forward strand as a template, and the strand that is used as a template will not be amplified, but the opposite strand to that template will be amplified. So when this forward inner primer comes and attaches to the reverse strands, the forward strand will be amplified. This is another DNA sequence that had the C nucleotide, the forward strand and G nucleotide, and the reverse strand. Now the primers comes and attached to the forward strand. When attached to the forward strand, this primer is the reverse primer, and it will get attached to the forward primer. It should be kept in mind, the reverse primers get attached to the forward strand, and forward primers get attached to the reverse strand, because the strand formed are opposite by the DNA polymerase enzyme because of the complementarity. So when it gets attached to the forward strand, so then it will amplify the reverse strand. This is the reverse inner primer specific for the CLE. It is specific by the C allele because it has a G nucleotide in the end, and this G is not able to get attached to the T nucleotide. So it will only work if T nucleotide is present. Similarly, the forward primer will only get attached if there is a presence of A nucleotides, and that for inner primer will not get attached to the C nucleotide or the G nucleotide because T only attaches with the A. Now we can see the nucleotide sequence in this figure. So this is the flower primer, and it will get attached to the strand only if there is C nucleotide present. If C is not present, then the G nucleotide will not be able to attach to this point. This figure highlights the mismatch that is intentionally introduced into the primer so that the specificity of the primer is increased. If the end nucleotide is not matching with the DNA sequence we want to amplify, and there is also the presence of another mismatch highlighted in this figure by the arrow, then there is no possibility that this DNA sequence will be amplified, meaning that we will have no false positive results. So the PCR will become very specific. This mismatch is intentionally introduced. We have another scenario that the primer is not able to get attached because there are two mismatches in this primer. First, the end nucleotide is not matched with the nucleotide we want to amplify, we want to screen in the DNA sequence. Secondly, there is a mismatch in every primer. When these two mismatches are present in the primer, then there is no amplification at all. In a second scenario, we can see that there is a presence of a mismatch, but it will not hinder the attachment because the end is getting attached. So this figure shows us how primer works and how primer do not work. The first primer will not work, and in the second figure the primer will work. The second primer will form a product and the first will not form a product. This is the DNA sequences, the primer, and that is another DNA sequence. The primer tries to get attached. This is a single primer and it will try to attach to both DNA sequences because it is present in both the DNA samples or in both the DNA PCR tubes and in every PCR tube. So it will try to attach to the DNA sequence. But let us see which primer gets attached and to which prime DNA it is able to attach and to which DNA it will not get attached. One DNA has a T allele and the other has a C allele, and this primer will try to attach to both DNA molecules. At the first end of the primer, we see that the G is complementary to C and it will get attached to this. But in the first DNA sequence, we can see that the G is not complementary to T and there will be no binding between G and T. So this is the first criteria which is shown in both the DNA molecules. In the first DNA molecule there is no binding because there is no complementarity between G and T, and in the second DNA the binding is successful because G is complementary to C. Now we know that we introduce mismatch in the primers, in the inner primers. So these, in the first figure we see that the mismatch is not able to find a bond with C. T and C bond is not forming. And this is similar in the other primer as well, because this DNA sequence is similar at other points but is only different at the point having T and C allele differences. The other nucleotides are complementary and will bind accordingly. We can see shown by the tick marks that there is successful bonds between them. Now you will see that in the first figure the amplification will not occur. In the first example the amplification will not occur because there are two mismatches in this DNA molecule with the primer we are trying it to attach, and this primer will be able to attach to the second DNA molecule and amplify it because it has only one mismatch, and that is also not present at the end of the primer but in the middle. And this will work as shown by the tick mark. So this is how a primer works, and this is how a primer does not work. This is the same primer amplified to different DNA molecules. One DNA molecule is not amplified, but the other DNA molecule is amplified. Applications of T-ARMS PCR. It is used for case control studies. It is used for genetic mapping, disease association studies, molecular breeding, and genome-wide associations. Now let us see, we have performed a T-ARMS PCR and we want to know what are the results. Then we will run the product of the T-ARMS PCR on an agarose gel. When we run it at 2.5% or 3% agarose gel, we will get this type of bending pattern. In this figure, the red bands show the base of the product of the outer primers, which was 446 base pair. The yellow bands show the product of the primer specific to the T allele, and the greenish bands show the product of the primer specific to the C allele, and these bands are formed on the basis of the length, and we know the product of each primer. So on the basis of the length of this product, we will be able to recognize which primer has worked and which primer has not worked. For example, in the bill or in the bands, having 3 bands, we can see that outer primers have worked and the 2 inner primers have also worked. It means, and so as a result 3 bands are formed. It means that this individual or this DNA obsolete from an individual has both the T and C allele, meaning that this individual is heterozygous. One chromosome of this individual has a T allele and the 585 microRNA gene, and the other chromosome of this individual contains the microRNA 585 gene having the C allele. So this is a heterozygous having the CT alleles. In the middle of the gel, we can see that there are only 2 bands. The first band is the product of the outer primers, and that is a base, and it is 446 base pair, and the other band is the shorter band, the greenish band, which is specific to the C allele. It means that both the chromosomes or both the genes of this individual had C allele on in them, meaning that this individual is homozygous for the C allele. So hence his genotype is CC genotype. In the right portion we can see that there are two bands. The first is the base band of the outer primers, and then is the middle band form. The primer having the middle length is formed, or the primer whose product length is intermediate between the other two primers. So we can see that this individual have the allele T. We can say this on the basis of the length of this yellow band, which was the length of the primer specific to the T allele. And as there is no presence of the other band, it means that this individual had T nucleotide on both the genes of microRNA 585 in his or her genome. So this is how we analyze and interpret the results obtained from ARMS PCR on an agar gel. We should keep a record of these results after we perform electrophoresis. So these arrows shows the genotypes of the DNA molecules for which we have carried the ARMS PCR. Now further elaborate this picture. This is the picture of a gel we have performed after a ARMS PCR. The well on the most right hand shows the DNA marker or gene ruler, which is used for finding out the length of the DNA molecules and the agarose gel. On the basis of this, we can see that there are three types of product. The first product is 426 base pair of the outer primers. The middle product is 297 base pair, and the outermost is a product having 202 base pair lengths. The yellow product is the formed by the inner forward and outer reverse primer amplifications. So on the basis of this we can know the different types of alleles in a gene in an individual. Now let us see how this works on the DNA molecule. Suppose this is a DNA molecule to which two primers are attached. These primers are known as the outer primers. This figure shows the outer primer attached to the DNA molecule. The second pore shows an outer primer attached to the reverse chain and an inner reverse primer attached to the forward chain, and the dotted red line shows the DNA region that will be amplified. meaning the length of this product. This is another DNA molecule having the T allele, to which two primers will get attached. The outer reverse primer, which will get attached to the forward strain, and the inner forward primer that will get attached to the reverse strain. And the black dotted rectangular shape region shows the region amplified by these two primers. Hence, on the basis of this, we will recognize the product size. Thank you for your time. For more videos, please subscribe to our channel Curious Minds. Thank you so much. Please subscribe to our channel Curious Minds.